Okay, we got just 6 slides here, you guys, on the chemical senses, taste and smell. So you have about 10,000 taste buds on your tongue. Mostly they're at the front part of your tongue, but there are some at the back of your tongue. They are in between these taste papillae, and there are a couple of different types of papillae. You can see on the picture the fungiform papillae at the front of the tongue here, and then also the lingual tonsils here, and these are the circumvallate papillae. So for each taste bud, there are about 40 to 60 cells, and these are epithelial cells. Only a few of those are actually taste cells. And those taste cells have little tiny microvilli on the top of them, which, as we've talked about lots of times, what does microvilli do for you? Gives you more surface area. Lots of similarities between taste and smell. On this picture, you can see over here at the side, right in here, this would be one taste cell. And the little taste hairs are sticking out right here through this pore, okay, which is pretty hard to see. But you've got some supporting cells. Here's the taste cell right here. This whole thing is called a taste bud, okay, and a lot of supporting cells around that. They're being replaced all the time, just like your skin cells. They're epithelial cells, and so they're getting replaced just like any other epithelial cell will. So every 7 to 10 days, you're making new ones. If you damage them, they're going to be replaced fairly quickly. So you see some of these on your tongue, little tiny bumps that you see on the tongue. We used to do taste bud mapping or taste mapping on our tongues, and they've decided now that really there isn't a taste map, but you might still see that on sites if you look up taste. And we used to just have these 4 that we talked about: sweet, salty, sour, and bitter. Anything that's got sugar in it or artificial sweeteners. Phenylalanine, for instance, is an amino acid that's an artificial sweetener. Saccharin is an artificial sweetener, are going to taste sweet to you. The bad part is that lead salts and things that are sometimes dangerous can also sometimes taste sweet. That's why little kids will, pick on old paint in old houses that has lead in it, and then they'll put their fingers in their mouth and they'll continue to do that because they like the taste that that lead gives them. Anything with inorganic salts in it will taste salty. Anything with hydrogen ions like lemons, oranges, any kind of citrus fruit, and other things, vinegar, will taste sour. And most of the things that are bitter are going to be things that animals won't eat, because a lot of bitter things are not good for you or dangerous, like morphine, nicotine, strychnine, but also some non-harmful things relatively, not caffeine. But a lot of medications that we take are bitter tasting. Don't put these at the back of your tongue because there are taste buds back there. You're actually better off putting them kind of in the center of the tongue. There aren't a lot of taste buds in the center, and then quickly swallow them with water or put them towards the back. I said not the back. I meant don't put them on the tip of your tongue because that's where you taste it the most. This new category called umami is probably only in the last 5 years they've decided that this is another taste category, specifically for one amino acid that's called glutamate. So we don't do taste bud mapping, but you might see that in other books. And again, if you look up on the internet, you probably will see taste maps of the tongue. So very similar to smell, and you did this in lab where you dried someone's tongue off and they put sugar on their tongue, and it took them maybe 30 seconds or a minute before they could taste it, because until it can be dissolved, it's not going to be able to be tasted. That there are different taste buds for different tastes. Obviously, the 5 categories each have a different type of receptor. You are least sensitive to things that are sweet and salty, most sensitive to bitter. That's probably because a lot of those things can be dangerous. And our taste adapts fairly rapidly. Think about putting a piece of candy or a piece of gum in your mouth, or even, you're really hungry and you go to eat some food, it tastes really, really good the very first few bites that you take, and then the taste is not nearly as obvious to you after you've taken several bites. Unless maybe it's something that you really hate. When we were talking about cranial nerves, we talked about some of the nerves that serve the tongue and are part of the gustatory pathway or the taste. A lot of this comes from the facial nerve. That's one of the biggest nerves in our cranial nerve group. And most of our taste buds are on the anterior part of our tongue. But there are a few in our back of our tongue, our throat. The pharynx is the throat. So glosso means tongue, pharyngeal, throat. So on the back side of the tongue and throat a little bit, there are some. And then also the vagus, further back in the pharynx, and also on your epiglottis, which is at the back of your throat. But taste is really influenced by a lot of other things. If you can't smell, you can't taste, because 80% of our taste is smell. But we also either like or dislike things based on things like texture of the food. You know, somebody might not really mind the taste of grapefruit, but really just hate the stringiness, or they might not mind the taste of banana, but really hate the texture of the banana. Pain receptors come into play when you eat really spicy things, and that can affect, and some people like that, some people don't. Kind of depends on what you grew up with. Smell is very similar to this, but obviously in your nose. It's way at the very roof of your nasal cavity. So if you think about it, it's pretty hard to get air up into the back of your nasal cavity, so you might have to sniff something, you know, really sniff in hard in order to be able to smell it if it's not very strong. Remember when we talked about the olfactory nerve, when we talked about the cribriform plate, and the olfactory bulb will have these little sensory receptors, nerve endings, bare nerve endings, bipolar neurons that come through that olfactory, from the olfactory bulb through the cribriform plate down into your nasal cavity. That's what all those little holes were for. These are the only neurons that are replaced throughout your life. They're not epithelial cells, they're neurons. So every 60 days or so you make new cells, and as we get older that slows down, and so our sense of smell gets less as we get older. There are probably around, there's a lot of debate about this, but around 7 major smells and at least 1,000 smell genes. Animals are way more sensitive than we are. They have a lot more surface area to their, and a lot more olfactory nerve endings. But for any of us, again, just like taste, things have to be dissolved, and that's why we have mucus in our nose. It has to be a water-soluble gas in order to dissolve in that mucus, because like all body fluids, it's mostly made of water. And then takes that information back up to the olfactory bulbs and then back to the olfactory cortex. But also remember that smells are really linked to the emotional system of our body, of our brain. And so it goes to the limbic system as well. And this is how, you know, you smell something and then that makes you remember a particular activity that maybe you haven't thought about for years, or a person, just based on what the smell was. Sometimes things will happen and people will get injuries that makes it so they can't smell. And that's called anosmia. So it could be damage to the head where they've damaged their olfactory nerves, or in most cases it's something like inflammation, obstruction of the nasal cavity, or just simply getting old. Zinc is needed for normal olfactory receptors, and so if you don't have enough zinc in your diet, you could gradually lose your sense of smell as well. But that's easy to fix by just simply replacing that, and then it would take a while, a few days or a week or so, to replace some of those cells. Okay, one question for you to think about, and then we will be finishing up the eye, starting on the ear on Monday. Okay, and the answer to that one is number 1. Doesn't have anything to do with your lungs, and they're not neurotransmitters. They bind to receptors, and just like any neuron conducting action potentials, it's opening the ion channels that stimulates those action potentials. Okay, I'll see you guys tomorrow.